Hello, I am Dr. Vijay Singh on behalf of Dr. Amit Rao. I am with you to discuss about an introduction to tissue culture technique with their scope and alternative source of phytopharmaceuticals. This chapter is to give you a concise knowledge. It shall try to induce your curiosity for the subject. I will deliver very brief on the basics of plant tissues culture and its application in production of phytopharmaceuticals. The subject of plant tissue culture is very vast and describing its every aspect is beyond the scope of this chapter. The readers are advised to dig into reading materials that is available online and offline to quench their thirst for comprehensive knowledge and information on subject matter of their interest. Now proceeding for module 3. About phytopharmaceuticals. Metabolism is a common function in living beings. The metabolism of carbon, nitrogen and some amount of other materials along with energy result in production of large amounts of carbohydrates and relatively smaller amount of lipids, proteins and nucleic acids. These are essential for overall function of the whole organism. They are therefore called primary metabolites. This is common phenomena for both plant and animals. However, in plants, all of the starting material of metabolism is not converted into primary metabolites. A large portion of them and the products of primary metabolism undergo secondary metabolism, which result in synthesis of a variety of organic molecules. These molecules are termed secondary metabolite. Secondary metabolite usually produced in very low amounts and their presence in plant is at times unique to a specific family, genera or even species. They usually do not play a direct role in normal functioning of cells. Initially, the specific role and function of such compound were not known. With the development of plant and biological sciences, the functions of such molecules could be understood. The more important are mentioned hereunder. They guard plant from insects, animals and microbial pathogens, save themselves from the harmful effects of UV light, air pollutant and other physical and chemical threats that occur due to different environmental conditions. The chemicals produce some special odor, color and taste to attract birds, insects and animals that helps in pollination and dispersal of seeds. Some phytochemicals are produced in reaction to an external stimuli, like when in danger. Secondary metabolism is also applicable in agronomy. In this field, the products of secondary metabolism play important function like increasing the germination time of spores till a favorable environment for growth exists, protection of spore in its different developmental stages from eating up by amoeba, eliminating microbe, insects and other species that can compete with it for air, water, sunshine and nutrition from its immediate surrounding during germination and growth. Humans have been harnessing this secondary metabolite phytochemicals or phytoconstituent for use as medicine, flavoring, colorants, repellents, recreational drugs and a variety of other purposes. The phytochemicals used in medicine can be specifically termed as phytopharmaceuticals. Now, production of phytopharmaceuticals in plant tissue culture. The scientists initiated the use of plant cell culture as an alternate source of research and production of secondary metabolite. at the end of 1960s. Today, number of medicinal plants are in use for establishing them in a variety of tissue culture systems for production of phytopharmaceuticals. Many of such plants fall in category of endangered or threatened species. Plant tissue culture is gaining popularity for producing phytochemicals of pharmaceutical importance at present time. Its use for the production of phytoconstituent of commercial interest has become significant since the introduction of sequonin produced by using cell culture of Lithospermum. Erythrogone in Japan, the metabolites that are being produced at present belongs to the category of agrochemicals, pharmaceuticals, nutraceuticals, food additive, etc. Like other mode of production, these techniques also has its advantages and limitations. The major ones are mentioned here under. Advantages: production conditions can be manipulated according to the requirement. The whole system is away from environmental, seasonal and geographical effects as well as soil conditions. The desired phytoconstituent can be produced consistently. Secondary metabolism can be synchronized to get optimum production of desired compound. It allows obtaining constituents from such species that is difficult to cultivate. The culture is free of pests, microbes and diseases. The cultured cells can be employed to produce new compound by applying genetic engineering and producing mutant cells. Production time through tissue culture is less and it is continuous. The phytochemical produced by cells can be extracted from the system quite easily. The production is free from contaminants like herbicides and pesticides. Now, limitation. In a large number of cases, the percentage of production is less. The undifferentiated cell culture are unable to produce a variety of compound as in plants they are produced by different organs. The genetic stability of cultured cell is under question as they are susceptible to undergo mutation. The mechanical system in the culture equipment may at times damage the cells. The manpower employed should be highly trained to handle the stringent aseptic environment and sophisticated instrument, making this process very costly. Aseptic conditions of the cell culture unit has to be flawless, otherwise the cell will get contaminated and deteriorated. Now, strategy developed to increase the yield of phytopharmaceuticals. The limitation listed above has restricted the commercial utilization of plant tissue culture. However, this technique has seen tremendous advancement in last 6 decades. It has also got support from development in genetic engineering and advancement in biological science. To overcome the limitation in the production of secondary metabolites, number of strategies has been developed and some of them are listed below. First is to employ genetically engineered selected cell line able to grow fast and give highly yield of desired compound. Second, to block biosynthetic pathway that interferes the production of desired metabolite. Third one, to inhibit the pathway that leads to degradation of the compound. Fourth, to stimulate the genetic responsible for the biosynthesis. Next is to employ combinatorial biosynthesis by introducing crucial gene into microbes. Next, to manipulate the medium. Next, to improve bioreactors for large scale culture. Next, to employ mechanism to inhibit the absorption of metabolite produced. Next is technique used in commercial production. Permeabilization. In plant cell culture, the products of interest that they produce remain inside the cells. They are blocked by the vacuoles present in the cell. Certain techniques have been developed that does not change viability or biosynthetic capability of cells, but overcomes the permeability of cells. This makes the cell to release phytochemicals in the media, which can be then easily collected. Charcoal is commonly used to absorb the constituent. The permeability can be induced physically as well as chemically. The physical agent commonly employed are sonication, electric pulse, UV radiation, etc. The chemicals commonly used are chitosan, DMSO, agarose, alginate and some organic solvent. In any case, care should be taken so that the viability and biomass is retained. Next is immobilized cell culture. Immobilization of plant cells helps in maintaining high level of biomass for long duration. This increases the productivity and at the same time reduce cost of production. Cells, their aggregates as well as callus can be immobilized physically, immobilized by entrapment. The entrapment can be done by one of the following method: gel entrapment. Gel like alginate, agarose, carrageenan, etc. are used alone or combination to entrap the cell by formation of ionic network, precipitation or by polymerization. Net or foam entrapment perform structure are used for entrapment. These structures are formed by cotton fiber, fiberglass, polyurethane and polyester fiber. Hollow fiber membrane entrapment. These are also performed structures that are formed by cellulose acetate, silicon polycarbonate. Now, culture of differentiated organ. The biggest drawback of cell culture is that they are in several instances unable to produce metabolite like entire plant. It is because the cell remain undifferentiated. Therefore, cultures of root or shoots are used and they have been found to be capable of producing metabolite satisfactorily. In this respect, hairy root culture is very successful, employed for commercial production of many compounds and also recombinant protein by using the genetic engineering. Hairy root culture. These cultures show high growth rate as well as genetic stability. The ability of hairy root cultures to synthesize metabolite is found to be higher than the roots. Hairy root is a cancerous disease of root which is caused when it is infected with a microbe. Agrobacterium rhizogenes. The other unique properties of hairy root culture are lateral branching, in large numbers abundance of root hair. Hairy roots are genetically stable. They show biochemical stability leading to higher production, absence of geotropism, condition of the medium is not required. Due to this property, the ability of hairy root culture is equal or more than the related plant root for production of secondary metabolite. Hairy root culture therefore have been utilized for production of compound obtained from root. Now, biotransformation. It is a process in which modification of functional group of the plant metabolite take place in the living cells. It is a chemical reaction that take place due to some catalyst. The result in production of compounds that are different from that produced by the normal metabolism. The plant cell have the ability to catalyze the reactions stereospecifically and this help in obtaining products that are chirally pure. The biotransformation reactions that take place in the cell are not possible through chemical or microbial synthesis. A wide range of reactions can take place in the cell like oxidation, reduction, acetylation, methylation, demethylation, etc. Biological catalysts like enzyme or phytoconstituent are now frequently employed to induce biotransformation in mass production of novel compounds through plant tissue culture. A successful biotransformation process required following conditions to be fulfilled: presence of essential enzyme in culture, agent employed should not be toxic to the cell culture, substrate should reach its target cellular compartment, the rate of further metabolism of the target product should be retarded. Now, techniques of biotransformation. Biotransformation by immobilized cell, biotransformation by hairy root culture, biotransformation by free cells, stimulation of chemical production. Some processes have also been developed that employs use of external stimulation to enhance the rate and quantity of metabolite production. Elicitation: it is a process to induce stress or defense related response in plant. This response prompts them to synthesize compounds at a higher rate as it does when it comes under attack by pathogens or sudden environmental stress. Elicitation improves the efficiency and production by decreasing the time. Change of media is not required. Enzymes involved in biosynthesis are stimulated. The secondary metabolites are directly excreted into the medium. Mechanism of elicitation. Elicitor signal effect is a complex activity that involves multiple component networks. It can initiate a range of reactions to defend itself. These reactions constitute some parallel or cross-linking signaling pathway that may result into responses at different targets. Plant signal molecules like reactive oxygen species, hydrogen peroxide, jasmonic acid, salicylic acid, ethylene, nitric oxide, etc. are responsible for stimulating the defense mechanism. All elicitors do not behave in the same way. The response may be different depending upon their organ, specificity, concentration, phytochemical environment, stage of their growth cycle, nutritional uptake, etc. Many mechanisms has been suggested in this regard. However, a set of sequence of activity that commonly take place in response to an elicitor can be as follows: binding of elicitor to the specific receptor that may be located on the plasma membrane, reaction between elicitor and receptor, generation of signal to activate nuclear gene responsible for plant defense, resulting in biosynthesis of specific phytocompound. Some of the effects that are exhibited due to elicitor are changes in calcium ion metabolism, membrane polarization may decrease, extracellular increase of pH, production of ROS and hydrogen peroxide contributing to the generation of bioactive fatty acid derivative, ROS involvement in the cross-linking of cell wall bound proline rich protein, accumulation of defense related and pathogenesis related protein, huge deviation in plasma membrane components. The physiological function like respiration, metabolism, ethylene production and peroxidase activity changes drastically. The genes involved in defensive mechanism is excited, which further stimulate enzyme and catalyst responsible to produce defensive metabolites. Classification of elicitor. The elicitor can be classified in two categories: Biotic and abiotic. Biotic elicitor. These are of biological origin, produced inside the plant or a pathogen. Chitosan polysaccharide, yeast, fungus, etc. are some type of biotic factor that have been used as elicitor in plant tissue culture. Abiotic elicitors. These are of non-biological origin. They belong to physical and chemical nature. Metal ion, organic, inorganic salt, radiations, heat, etc. are some example of abiotic elicitor. Precursor feeding. In this process, certain intermediate compound occurring in biosynthetic pathway of target metabolite is fed into the system to increase the reaction rate. On addition of the intermediate, the enzyme responsible for the further biosynthetic step are initiated and biosynthesis is accelerated, resulting in higher product concentration. However, the concentration of such intermediate should be optimized. At higher concentration, it can inhibit the enzyme as well as growth of cells, and at low concentration the desired activity is not achieved. For every plant species, the precursor concentration has to be properly worked out. The technique has been used in number of cases in commercial production to improve the yield of target compound. Now, bioreactor for production of secondary metabolites. Bioreactors are the mechanically manufactured holding device in which the environment can be controlled and maintained to allow large scale cultivation of plant cell cultures. Different type of bioreactors has been designed and engineered for the production of secondary metabolites. They are termed as batch, continuous, multi-stage, airlift or stir tank bioreactors. Bioreactors employ any one or a combination of above mentioned technique to maintain different type of cultures for continuous and large scale production of phytochemicals of pharmaceutical interest. However, there are some binding factors that need to be kept in consideration while employing and operating bioreactors. There should be continuous and sufficient supply of oxygen. The aeration and agitator speed should be controlled so that the cell in the culture are not ruptured. The process parameter like nutrition, temperature, light, etc. should be optimized to keep a steady state of growth. Now, Module 4. Conclusion and summary. Plant tissue culture exploits the capability of any plant part or a tissue to survive or proliferate if it is grown in controlled environment and in a media that has sufficient nutrients for it. The culture can be initiated from any plant part. The requirements of nutrition and media differ from species to species and also at times from plant part to plant part. In same species, plasticity and totipotency are the two important central points of plant cell culture. Plant tissue culture started developing as a science when cell and cell theory come in existence. There are mainly two major techniques in plant tissue culture: static culture. In this procedure, the plant tissue is grown on solid agar medium, whereas in suspension cultures the development and growth take place in liquid media. The suspension culture are of two types, mainly batch suspension cell culture where growth take place in fixed volume of nutrient medium, and continuous suspension culture. In this, the media is continuously changed. The different type of culture are root culture, flower, ovary and ovule culture. Culture of shoot, species and leaves embryo culture, endosperm culture and other pollen culture and protoplast culture. In 1909, Kunster isolated protoplast by mechanical method. A representative nutrient medium for plant tissue culture consists essentially of components like inorganic, macro as well as micro element, carbon and energy source, vitamins, amino acids, growth regulator or plant hormones and solidifying agent. Some undefined supplements of organic nature are also added like casein hydroxylate, variety of organic extracts, ground banana, coconut milk, malt extract, yeast extract, tomato juice, activated charcoal, etc. may be added for specific purpose. An optimum pH of 5.7 is vital for cultured cell. Composition of plant tissue culture media depend on number of things. The important among them are plant species, material used and purpose of culture. Amongst the various composition used to formulate media, MS medium is the most frequently used due to its versatility. The basic laboratory process involve preparation of stock solution of growth regulator, preparation of nutrient medium, sterilization of medium and equipments, micropropagation of plantlets that usually has 4 distinct steps which are performed one after the another, and finally plantlets are generated that are then transferred to greenhouse and subsequently to the field. Plant tissue culture has application in fundamental research and also in commerce. It is widely used in agriculture, forestry, horticulture and also in the field of pharmacognosy. In the field of pharmacognosy, tissue culture is employed for cultivation of medicinal plants that are endangered and extinct. Its other application is in the field of producing phytopharmaceuticals. Plant tissue culture is gaining popularity for producing phytochemicals of pharmaceutical importance at present time. Its use for production of phytoconstituent of commercial interest has become significant since the introduction of ciconin produced by using cell culture of Lithospermum erythrosum in Japan. However, this type of production has its advantages as well as its limitations, but there has been unparalleled advancement in last 6 decades. It has also got support from development in genetic engineering and advancement in biological science. To overcome the limitation in production of secondary metabolites, number of strategies. Those strategies are now being applied with techniques like permeabilization, immobilized cell culture, culture of differentiated organ like hairy root culture, biotransformation, elicitation and precursor feeding for improving the yield of phytopharmaceutical in commercial production. Biological catalysts like enzymes or phytoconstituents are now frequently employed to induce biotransformation in mass production of novel compounds through plant tissue culture. Different type of bioreactor has been designed and engineered for large scale production of secondary metabolites. Plant tissue culture, after being incorporated with plant transformation, has resulted in growing number of species that are being genetically improved with strong resistance from pests and pathogens. This is helping in great deal in agriculture, forestry and horticulture, especially to grow some exotic species. Similarly, invention and new innovation are making it possible to produce phytopharmaceutical in large scale, and it is expected that In coming year, many more novel compounds will be produced in our evolving bioreactors. Learning outcome: by the end of this course, the student will be able to appreciate and convey the basic concept of plant tissue culture, gain expertise in aseptic technique, design and perform experiment in lab, innovate in the field of plant tissue culture, set up an experiment with plant species of their own choice for production of phytopharmaceuticals, comprehend and control different parameters for production of phytopharmaceutical, set up a lab for research or commercial purpose. Part A, B study module 1 and module 2. Part B, B study module 3 and module 4. Dear student, hope you enjoyed this episode. Keep learning. Goodbye.